Hello, my name is Joshua Mugabe. I welcome you to Seed Structure. A seed is the product of sexual reproduction in flowering plants. So our seed here, it is a product of sexual reproduction in a flowering plant. Here we have our seed, and it has an outer cover, which is the tester. We have the middle part, that one is the cotyledon. We have the upper part of the embryo, that one is called the plumule. Then the lower part of the embryo, we call it the radicle. Then there is a small hole in the tester, that one we call it a micropyle. In dicotyledonous plants, the seeds consist of a miniature plant, the embryo, and two modified leaves, the cotyledons, swollen with food reserves. So we have the dicotyledonous plant seeds, like the ones we are seeing here. This is half a cotyledon. These are two cotyledons with the tester removed. They consist of a miniature plant. A miniature plant is this one we have, like the embryo. It is a plant, but very miniature, very small. And it is the embryo. Then it has the embryo, which of course is this one, our miniature plant, and the two modified leaves. The two modified leaves we are talking about are these ones, which we call the cotyledons. These ones are modified leaves. They are modified leaves, and those ones are swollen with food reserves. Those are our cotyledons. The tester. The tester, this is a seed coat. It encloses and protects the seed from insects and fungi. So we have that part which we are calling the tester. It is this outer covering. This outer covering is what we call the tester. And its main function is to protect and enclose the seed from insects and fungi. The tester encloses and protects the seeds from insects and fungi. It is also called a seed coat. It is usually hard and dry. The tester is very hard. That's why it is functioning as a protector. It is very hard, and it is always dry. It is always dry. We have a micropyle. A micropyle, this is a hole which admits water when the seed starts to germinate. That is our micropyle. The micropyle is this small hole here. That is our micropyle, and its main work is to allow entry of water. So water will enter through the micropyle and it will enter into the cotyledon. So the micropyle allows the entry of water into the seed when the seed starts to germinate. We have the plumule. The plumule, the embryo shoot. This is the embryo shoot. It has 2 leaves and a growing point. That is the plumule. So the plumule is this part that we are seeing here. A plumule has 2 leaves. It has 2 leaves and it has a growing point. It has got a growing point. So that one is what we call the plumule. The cotyledons. These are modified leaves containing food reserves. Those are the cotyledons. The cotyledons are these ones. This is a cotyledon, and the other one, the other side, is also a cotyledon. What do they contain? They contain food reserves. They are leaves, but we call them modified leaves because they are storing food. That's why we are calling them modified leaves with food reserve. We have the radicle. The radicle is the embryo root. It is the embryo root. This one here is our radicle, and it is part of the embryo. So the embryo has a root, and that root we call it the radicle. We have the epicotyle. Epicotyle, this is the section of the stem above the cotyledon stalk. It is the section of the stem above the cotyledon stalk. The cotyledon stalk is this one, and this is the section here of the stem above the cotyledon stalk. Epicotyle, epicotyle, above the cotyledon stalk. So here we have our epicotyle part, the section of a stem above the cotyledon stalk. We have the hypocotyle. Hypocotyle. Hypocotyle, this is the section of the stem below the cotyledon stalk. Below the cotyledon stalk. So here we have the cotyledon stalk, but that part of the stem below the cotyledon stalk is what we call the hypocotyle. That section of the stem below the cotyledon stalk is what we call the hypocotyle. Hello, my friend. Thank you very much for participating in seed structure. Please do like, share, and subscribe.